Imagine a world where genetic diseases are a thing of the past, where parents can ensure their children are born free from hereditary conditions that have plagued humanity for millennia. A world where we can edit our DNA as easily as we edit a document on our computers. This isn't science fiction. This is the promise of CRISPR technology, but with great power comes great responsibility and great controversy. Welcome to the cutting edge of genetic engineering, where breakthrough science collides head-on with ethics, morality, and the very essence of what it means to be human. In this video, we're diving deep into the world of CRISPR and the shocking case of the world's first gene-edited babies. Buckle up, because this story has more twists and turns than a double helix. November 2018. The Second International Summit on Human Genome Editing in Hong Kong. The scientific community is rocked by an announcement that would send shockwaves around the world. Chinese scientist He Jiankui takes the stage and declares that he has created the world's first gene-edited babies, twin girls named Lulu and Nana. In an instant, the line between theoretical possibility and controversial reality is crossed. The future of genetic engineering is here, whether we're ready for it or not. Before he became a household name synonymous with controversial science, He Jiankui was a rising star in China's booming biotech scene. Born in 1984 in a small village in Hunan province, his journey to the forefront of genetic engineering is a testament to ambition, brilliance, and the rapid pace of scientific advancement in China. His early life was marked by academic excellence. He studied physics at the University of Science and Technology of China, where his intellect quickly set him apart. But it was during his doctoral studies at Rice University in the United States that his interests began to shift towards biology and genetics. After completing his PhD in biophysics, he continued his postdoctoral research at Stanford University. It was here, in the heart of Silicon Valley, that he was exposed to the entrepreneurial spirit that would later define his career. He wasn't content with just conducting research, he wanted to change the world. In 2012, he returned to China as part of the country's 1,000 Talents Plan, a program designed to attract top Chinese scientists back from overseas. He quickly established himself at the Southern University of Science and Technology in Shenzhen, a city often referred to as China's Silicon Valley. But academia alone couldn't contain his ambitions. He founded two biotechnology companies, Direct Genomics and Viennomics. These ventures focused on developing gene sequencing technologies and precision medicine, positioning he at the intersection of scientific research and commercial application. It was during this time that he became fascinated with CRISPR technology. CRISPR, short for Clustered Regularly Interspaced Short Palindromic Repeats, had burst onto the scientific scene in 2012, promising a revolution in gene editing. He saw in CRISPR not just a powerful research tool, but a way to make history. To understand the magnitude of He Jiankui's actions, we need to grasp the revolutionary nature of CRISPR technology. CRISPR is often described as genetic scissors, but this simple analogy belies its true power and potential. At its core, CRISPR is a gene editing tool that allows scientists to make precise changes to DNA. It's based on a natural defense mechanism found in bacteria, which use CRISPR to cut up the DNA of invading viruses. Scientists Jennifer Doudna and Emmanuel Charpentier realized this bacterial defense system could be repurposed as a powerful gene editing tool. A discovery that would earn them the 2020 Nobel Prize in Chemistry. The potential applications of CRISPR are staggering. 1. Medicine. CRISPR could potentially cure genetic diseases by correcting faulty genes. Imagine eliminating hereditary conditions like cystic fibrosis or Huntington's disease. 2. Agriculture. Crops could be made more resistant to pests, drought, and disease, potentially revolutionizing food production and security. 3. Environment. CRISPR could be used to create organisms capable of cleaning up pollution or to bring back extinct species. 4. Basic research. CRISPR allows scientists to study gene function more easily than ever before, accelerating our understanding of genetics and disease. But with this immense power come profound ethical questions. 1. Where do we draw the line between treatment and enhancement? 2. Who gets to decide which genes are good or bad? 3. What are the long-term consequences of editing the human genome? 4. Could gene editing exacerbate social inequalities? These were the questions swirling in the scientific community as He Jiankui began to formulate his audacious plan. In 2016, as debates about the ethics of gene editing raged in scientific circles, He Jiankui made a decision that would change the course of his life and potentially the future of humanity. He decided to push the boundaries of CRISPR technology further than anyone had before by creating the world's first genetically edited babies. His target was a gene called CCR5. This gene produces a protein that HIV uses to enter cells. A naturally occurring mutation, CCR5 to D32, can provide resistance to HIV infection. He aimed to recreate this mutation using CRISPR, potentially making the babies resistant to HIV. The experiment unfolded in several stages. 1. Recruitment. He and his team recruited couples where the father was HIV positive and the mother was HIV negative. He promised them a chance to have children protected from HIV, a significant health issue in China. 2. In vitro fertilization. The couples underwent in vitro fertilization, IVF, to create embryos. 3. Gene editing. His team used CRISPR to edit the CCR5 gene in these embryos. This was the crucial and controversial step, making changes to the human germline that could be passed down to future generations. 4. Implantation. The edited embryos were implanted into the mother's uteruses. 5. Monitoring. The pregnancies were closely monitored for any signs of complications. 6. Birth. In October and November 2018, twin girls, later given the pseudonyms Lulu and Nana, were born. Throughout this process, he operated in near total secrecy. His own university was unaware of the experiment. He didn't publish his research in peer-reviewed journals or seek proper ethical approvals. Instead, he prepared to shock the world with a fait accompli. The news spread like wildfire. Scientists, ethicists, and the general public were stunned. This wasn't supposed to happen. Not like this. Not yet. In his announcement, he claimed several justifications for his work. 1. Protection from HIV. He argued that the genetic modification could protect the children from HIV, a significant health issue in China. 2. Informed consent. He stated that the parents were fully informed and had consented to the procedure. 3. Ethical obligation. He believed his work would help millions of children, framing it as an ethical imperative. 4. Technological leadership. There was an undercurrent of national pride, positioning China at the forefront of genetic engineering. But the scientific community was far from convinced. Criticisms came swift and harsh. 1. Premature and reckless. Many argued that gene editing in embryos wasn't yet safe or well understood enough for clinical use. 2. Lack of transparency. He had conducted his experiment in secret, without proper oversight or peer review. 3. Questionable necessity. There are already effective ways to prevent HIV transmission from parents to children, making the genetic modification unnecessary. 4. Unknown risks. The long-term effects of this genetic modification were entirely unknown, potentially putting the children at risk. As he took the stage at the Hong Kong summit, he faced a barrage of questions from shocked and angry scientists. The world watched as the drama unfolded, and the debate over the ethics of human genetic engineering exploded into the public consciousness. Chinese government. Initially silent, soon launched an investigation into He's work. He was placed under house arrest, and his research was halted. As investigations proceeded, more troubling details emerged. 1. Informed consent issues. Questions arose about whether the parents fully understood the risks involved in the experiment. 2. Data concerns. The limited data he shared showed potential issues with the editing process, including mosaicism, where not all cells carry the intended edit. 3. Off-target effects. There were concerns about unintended edits in other parts of the genome, which could have unforeseen consequences. 4. Incomplete editing. Analysis suggested his team may not have successfully recreated the CCR5 to D32 mutation, potentially exposing the children to unknown risks without the intended benefit. The hammer fell in December 2019. He Jiankui was sentenced to 3 years in prison for illegal medical practice. He was also fined 3 million yuan, about $430,000, and banned from working with human reproductive technology for life. The scientific community breathed a collective sigh of relief, but the questions his experiment raised didn't go away. The genie was out of the bottle, and the world had to grapple with the reality of human genetic engineering. The ripple effects of He Jiankui's experiment extended far beyond his own fate, influencing scientific practice, policy, and public perception worldwide. The fate of Lulu and Nana, the gene-edited babies, remains a topic of concern and speculation. In 2019, China announced that the twins and a third gene-edited baby born later would be under state medical supervision for their lifetimes. The long-term effects of the genetic modifications on their health and development remain unknown. As for He Jiankui, he was released from prison in April 2022. His future plans and the full extent of his research remain unclear, but his name will forever be associated with this controversial milestone in scientific history. The story of He Jiankui and the world's first gene-edited babies is more than just a scientific scandal. It's a cautionary tale about the dangers of unchecked ambition and the profound responsibilities that come with groundbreaking technologies. He Jiankui sought to make history, and he succeeded, though perhaps not in the way he intended. His actions forced us to confront challenging questions about the future of our species and the ethics of rewriting our genetic code. Imagine again that world where genetic diseases are a thing of the past. It's a compelling vision, but how we get there matters just as much as the destination itself. If you found this video fascinating, don't forget to like, comment, and subscribe for more deep dives into the world of cutting-edge science and technology.